How does? Okay, good morning. We're going to get started with this session on demystifying the role of tumor immune environment in pancreatic cancer. We have four outstanding presentations, 15 minutes each, followed by we'll have the discussion at the end. So my name is Tian Ko. I'm from McGovern Medical School in Houston, and my co-moderator is Dr. Asapal from Cedars-Sinai. So our first speaker is Dr. Yan Cheng, who is an assistant professor in the Department of Translational Molecular Pathology in the Division of Pathology and Laboratory Medicine at the University of Texas MD Anderson Cancer Center in Houston. The title of his presentation is Deciphering the Pancreatic Tumor Microenvironment Using New Transgenic Mouse Models. Dr. Chen.
Thank you, Dr. Cole, for the kind introduction, and I really thank the organizers for their invitation. This is not my slide, I think. No. This is not my presentation. Right. I think this is the next session. So Yang Chen. So using this time, I will just start. Okay. Sure. Sure, thanks. Yep, this is mine. So it's an honor to present and to share with you some of our recent and ongoing work, and mainly focus on pancreatic cancer, but also some of our mouse models showed some phenotypes of pancreatitis. That's why I think the presentations here at this meeting are really, really helpful for my research. And speaking of tumor microenvironment of pancreatic cancer, it's a very complicated entity composed of both the cellular components and the extracellular matrix component. And when I was a postdoc in Dr. Raghu Kalluri's lab, who presented like 3 days ago in the evening of the first day, when I was in his lab, our long-term interests were associated with cancer-associated fibroblasts, which are, now we know, super heterogeneous with different functions and subtypes in the cancer. And speaking of fibroblasts, we know that they are the contributors to the fibrosis in pancreatic cancer. And the main reason of the fibrotic environment of pancreatic tumor and many other solid tumors is the superabundant deposition of type 1 collagen. As shown here, when we check the pancreatic cancer tissue slides, we see the majority of the tissue areas are actually covered by type 1 collagen. And but the function of type 1 collagen is controversial in the field, whether it's promoting or restraining the tumor progression. Partly because of the lack of specific cell type specific knockout mouse models for type 1 collagen. That's why when I was a postdoc, we developed several transgenic mouse models with type 1 collagen specific depletion in either fibroblasts or cancer cells in the context of spontaneous pancreatic tumor formation. And in my first cancer cell paper, I identified that specific depletion of the type 1 collagen from fibroblasts actually made the tumor worse, indicating that in some contexts, collagen from fibroblasts can actually restrain the tumor instead of promoting the tumor. On the other hand, we found that when we depleted type 1 collagen from cancer cells, we observed the opposite phenotype, that the tumor got better. That's why we check the collagen from either cancer cells or fibroblasts, realizing that the cancer cell actually made their own very unique abnormal collagen 1 subtype, which is absent in our normal body, which is the homotrimer format. And because we know that collagen 1 is composed of two chains, like two types of chains, the alpha-1 chain encoded by COL1A1 gene and COL1A2 gene encoding the collagen 1 alpha-2 chain. And in cancer cells, due to the DNA hypermethylation and this gene silencing of the COL1A2 gene, cancer cells only express the COL1A1 gene, leading to the alpha-1 chain forming the type 1 collagen homotrimer, which is abnormal and oncogenic. On the other hand, fibroblasts, as well as all the fibroblasts in our normal body, they express both COL1A1 gene and COL1A2 gene, leading to the synthesis of the normal format of type 1 collagen, the heterotrimer format. And when I established my lab 2 years ago as an independent PI in MD Anderson, I was thinking what we can build from what we already published. So in our previous studies regarding the type 1 collagen, I established a variety of transgenic mouse models with single-cell RNA sequencing databases. So now I have the opportunity to integrate all of these models, sequencing data, and cross-compare what might be the universal responding gene signature of the type 1 collagen homotrimer versus heterotrimer. So I screened for a variety of genes. I called them collagen 1 responsive genes. So for example, these set of genes are suppressed by the good collagen made by the fibroblasts. So the fibroblast-derived type 1 collagen restraining the tumor and suppressing these genes. On the other hand, type 1 collagen made by the cancer cells, the bad collagen 1 homotrimer, they can upregulate this same set of genes, meaning that these genes are potentially the downstream mediators of the collagen 1 homotrimer heterotrimer balance in this disease. So next, when I established my lab, I purchased those available flocks or knockout mouse strains from JAX. LGALS3 encoding the galectin-3 is one of our top candidates because when we knock out type 1 collagen in cancer cells, when the tumor got better, we saw significant decrease of galectin-3 in the tumors. So first, we analyzed the human data set from either the Chinese data from the published in Cell Research, also as well as the NINAS data published in Nature Cancer. Both are human pancreatic cancer. We see that galectin-3 is highly expressed mostly in myeloid cell macrophages, but they are also highly expressed by cancer cells. And also from the data, spatial data from the Dr. Annabelle Mitra's lab published in Cancer Discovery last year. And we can see that the galectin-3 expression pattern is generally overlapping with the cancer markers, such as the cytokeratin 8. Indicating the involvement of this protein significantly with cancerous lesions. And in collaboration with Dr. Huamin Wang, we examine a set of tissue microarray from human pancreatic cancer. We can see that the high expression levels of galectin-3 in human pancreatic patients are correlated with poor prognosis, and consistent results were observed from TCGA database that high expression level of LGALS3 was correlated with poor prognosis. So that's why we crossed the KPC mice with galectin-3 whole body knockout from JAX, because we used whole body knockout mouse because as I showed earlier, this protein is highly expressed by both myeloid cells, macrophages, and the cancer cells. So we want to delete all of them. And then we realized that the knockout of galectin-3 can significantly inhibit tumor progression. In order to understand why this happens, we utilize the single-cell RNA sequencing to compare the control tumors versus the galectin-3 knockout tumors. The first thing we notice is the significantly accumulated macrophages, as shown by the purple and the pink color. Especially as we compare the control tumor versus knockout tumor, we can see the significant accumulation of the purple color labeled subtype of macrophages. Further analysis revealed that this enriched myeloid cell one subcluster more resembled the M1-like MHC2 high macrophages. So they are usually considered as the good immune stimulatory macrophages in the solid tumors. And here we show that the MHC2, one of the genes is highly upregulated in the knockout tumors, together with downregulation of the immunosuppressive markers like PD-L1, arginase-1, VEGF-A, etc. And also we noticed that by flow cytometry and immunostaining, we can confirm the significantly accumulated MHC2 high macrophages. And also we can see that accompanied with more M1-like macrophages in the knockout tumor, we can see more T cells coming in the tumors. And also in TCGA human data set, we can see the reverse correlation between galectin-3 gene and the T cell genes. And next, we hope to see whether the increased T cells can be a window or opportunity for enhancing the immunotherapy response in pancreatic cancer in the knockout mice, because we know that pancreatic cancer is extremely refractory to all kinds of immunotherapy. And so that's why before combining immunotherapy with our galectin-3 knockout, we also identified whether we can find a secondary target that can be further combined into this therapeutic system. And that's when we noticed the Nature Medicine paper related to the anti-PD-1 and anti-CXCL12, CXCR4 axis. This preclinical and clinical study, they use this inhibitor for CXCL12, CXCR4 as well in combination with antibody against PD-1, achieving some promising results. And interestingly, we happen to notice that CXCL12 is one of the top upregulated genes when we now call galectin-3. Even though we know that galectin-3 knockout significantly inhibited tumor progression, but somehow due to some unknown reason, we observed that the CXCL12 level, either in serum or in the tumor or in fibroblasts from the tumors, the levels were significantly upregulated as potentially a host response due to the galectin-3 knockout. Anyway, we think that it might be a compensating mechanism after the galectin-3 knockout if we can target this signaling pathway by inhibiting CXCL12 interaction with its receptor CXCR4, maybe we can achieve better therapeutic outcome. And also in TCGA database, we can see a very clear reverse correlation between the galectin-3 and the CXCL12 in the pancreatic cancer patients, meaning that when a patient is galectin-3 low case, even though their survival is higher, but they will have more CXCL12. Based on this rationale, as summarized in this cartoon, we see that galectin-3 is highly upregulated in both cancer cells and macrophages in mediating their crosstalk to promote tumor progression. And on the other hand, if we can target galectin-3, we can increase the T cell infiltration and we can combine potentially anti-PD-1 therapy into this system. Also, because we also observe CXCL12 upregulation, so if we can target all of the three elements in this crosstalk, we may achieve better therapeutic efficacy. So it was indeed the case using the KPPC mice spontaneous mouse model of pancreatic cancer. We treated them with the combination of the therapies, and the triple combination achieved the most promising results. This paper was published earlier this year in gastroenterology as the first paper in my independent lab. And now I hope to spend the last few minutes to introduce another paper we recently published. It's a very different topic. We all know that in pancreatic cancer, oncogenic KRAS mutation is the key driver, occurring in 90% or above cases of the pancreatic tumor cases. But there are actually 10% KRAS wild-type patient, and they usually harbors different variety of combinations of tumor suppressor gene loss, such as P53, SMAD4, CDKN2A, TGF-beta receptor 2, etc. And when I established my lab, I was thinking that since all of the commonly used mouse models, spontaneous mouse models for pancreatic cancer in the field are based on KRAS mutation-driven pancreatic cancer, whether we can introduce a mouse model system that is independent of oncogenic KRAS. That's why in this model system, as I shown here, on the left is PPSSC. Without KRAS, we introduce the P53 loss and SMAD4 loss. On the right, we have the PPTTC model with the P53 loss in combination with the TGF-beta receptor 2 loss. Two major tumor suppressors like these are all the three major tumor suppressors in pancreatic tumors. And interestingly, they did develop spontaneous pancreatic tumors. And more interestingly, that the PPSSC with the P53 and SMAD4 loss not only introduced the spontaneous pancreatic tumor formation, but the tumor is a very unique and rare, relatively rare subtype. It's the adenosquamous subtype. Instead of usually the KPC mouse, they developed the classical adenocarcinoma subtype. As shown here, on the left is the KPPC model harboring KRAS and P53 loss. They develop a classic adenocarcinoma subtype. And on the right, I show the histology of the PPSSC model with the P53 loss and SMAD4 loss, and showing the very distinctive adenosquamous pathology. And we can confirm that after isolation and generation of the primary cancer cell line in culture from these mice, we can maintain their adenosquamous phenotype, as we showed from bulk RNA sequencing data. And if we inject those cells into a background matched Black 6 mice, they will continue to form tumors in pancreas, which can provide a syngeneic mouse model very unique for the study of adenosquamous pancreatic cancer. As shown here, after orthotopic injection into the Black 6 mice, they maintain their adenosquamous phenotype and the high expression level of cytokeratin 5 or 6 or P63. And it's interesting that there is one inconsistency between our mouse model and the KRAS wild-type patient case, because our mouse model predominantly developed this adenosquamous subtype. However, in the 10% of the KRAS wild-type pancreatic patient cases, 90% of them still get the classical adenosquamous adenocarcinoma subtype, but not the adenosquamous subtype. So it's something that we are studying. What would be the reason for why the KRAS wild-type mouse model predominantly developed adenosquamous subtype? And but there is one coincidence that we observe very interestingly that is related to the very commonly used BXPC3 cancer human pancreatic cancer cell line, because we do have one cancer cell line from human origin that is KRAS wild type, and this BXPC3 cancer cell happens to have the same genotype with P53 loss and SMAD4 loss, and more importantly, it has the adenosquamous subtype. So it matches what we observed in at least this cell line matches what we observed using our mouse model. In the end, I would like to thank my previous and current mentors, my collaborators and lab members, and the funding opportunities. Thank you.
Thank you, Dr. Chen. Our next presenter is Dr. Diana Martinez Saucedo from Yale University School of Medicine. Her presentation is Reigniting Immunity in Pancreatic Tumors, Delivery of RNAs Using Engineered Lupus Antibodies.
Thank you for that introduction. Thank you to the organizers for the invitation. I'm going to tell you how we are delivering therapeutic RNAs into PDAC tumors using a lupus-derived antibody. Oh yeah. So I'm going to take you back in time and to the first question that we have, and that was, how can we deliver RNAs into PDACs? So our collaborators from the Glaser lab were working with this lupus-derived antibody that has two important characteristics. It can bind to DNA, and the second one is that it penetrates into the cell. So they hypothesized that if they modify the nucleic binding pocket by expanding the positively charged, they could make it to bind also to RNA. So they made a single point mutation, and with that, they hypothesized that we will be binding to RNA. And sure enough, this mutation was enough to make the antibody to also bind to RNA, and not only that, but also protect it from degradation. So now that we have the tool, what RNA, therapeutic RNA, are we gonna deliver using this antibody? So among the options, they decided to go for the 3-PHP RNA, that is a short hairpin that mimics influenza virus, and that in previous studies has been described that it activates type 1 interferon response. So can we deliver this short hairpin using the antibody into cancer cells, pancreatic cancer cells in specific? So what we do it is that we have our cells and we treat them with the antibody plus the short hairpin, and look how the antibody is reaching the cell, and also, like most importantly at this point, is that the short hairpin is within the cell, showing that we were able to deliver this antibody into the antibody and the short hairpin into the cancer cells. So now, sure, we are reaching the cell, delivering the RNA, but are we really triggering the type 1 interferon response that we wanted? So to do so, in these previous studies that I was telling you about, the short hairpin has described to be recognized by RIG1, and that with that, the consequence is that RIG1 then activate type 1 interferon response. So what we did here is that we culture the cells with the complex and treat them with the complex or the single treatments. And we measure type 1 interferon response, and you can see how the complex increase the production of these type 1 interferons. And not only that, but we also went ahead and did a knockout for RIG1, and you can see how this effect is abolished. So probably at this point, you might be wondering, how is TMAP actually penetrating into cells? So again, this lupus-derived antibody was previously being described to engage with ENT2, and through ENT2 to penetrate into the cell. ENT2 might be ringing a bell into you because this is one of the transporters that is also used by gemcitabine. So we know that this transporter is highly expressed in pancreatic cancer cells. And just to test that the mutation that was made for TMAP development was not altering the way it was transported into the cell, we again treat the cells with different inhibitors for ENT2. And you can see here clearly how we lose the penetrating activity of TMAP3. So up to this point, I have shown you how the complex TMAP3 and the short hairpin are entering into PDOC cells and how they are triggering the type 1 interferon response. But are we going to be able to deliver in PDOC cells in vivo? So what we did is that we established the orthotopic KPC model. And after 10 days post-implantation, we injected intravenously the label TMAP3. 24 hours after, we found that the TMAP3 is located specifically to pancreas. So we went ahead and repeated this experiment. And this time, 24 hours after, okay, so the treatment was like the antibody plus the short hairpin. So 24 hours after the treatment, I went ahead, took out the pancreas, dissociate the cells, and sort them into tumor and immune cells. And I was surprised to find that I was able to find the expression of the short hairpin specifically in the tumor cells. And I even remember Luisa asking me, like Dr. Escobar asking me about these results, and I put a hold because I wanted to repeat them. And sure enough, I got the same result. So I was like, okay, this is what we have. So now that we know that the antibody was reaching the pancreas and the short hairpin is being delivered directly into pancreatic cancer cells, can we improve animal survival? So again, we established the orthotopic KPC model, and this time we give 3 treatments 10 days post the implantation. And you can see here how of all the groups that we have with the treatment, with the treatments with the single controls, only the ones that were treated with the antibody and the short hairpin had an increase in their survival. And we know that these type of tumors are very aggressive. So we were very happy to find that we were seeing an effect. So is this effect dependent on activating the type 1 interferons in the pancreas? So what we did next is that we took the tumor cells, and in them, they look for genes that were associated with RIG1. And we found that there was an increase on the expression of these genes when the antibody and the short hairpin were used to treat the mice. But we also wanted to know if after the production of these type 1 interferons, the cancer cells were responding to it. So we look also for interferon-stimulated genes, and we see that, again, just in the group that was treated with the short hairpin and the antibody, we see increase in these genes, meaning that we are triggering the RIG1 activation and the response to type 1 interferons. So our next question was, does the type 1 interferon response triggered by our complex is activating immunity, anti-tumor immunity? So I'm going to skip most of what we did in this part because of time. But one important thing that we found is that T cells were highly expressed in the groups that were treated with the antibody plus the short hairpin. And not only that, but the T cells that were increased in this group were the CD8 cells. So to corroborate these findings, I did a Western blot, and I found that they were increased in the group that was treated with the TMAP and the short hairpin. And we also were able to corroborate this data using flow cytometry. But as I was telling you, we found that the increment was in activated CD8 T cells. So we measure the activation markers for these cells, and we saw an increase in the expression of these genes. And again, we corroborate using flow cytometry for CD8 and granzyme B. So the next step that we took to see the dependence of the TMAP and the short hairpin to T cells was to use RAC1 knockout mice. So again, we repeated the orthotopic model, and we give the 3 doses. And you can see here how the advantage that was given by the antibody and the short hairpin was completely abolished when the T cells were not present in the tumor microenvironment. So the last part of my talk, I want just to show you how specific is this response to induce the T cell activation. And to do so, what we did is that we had KPC wild type cells that were treated with the antibody and the complex, and the antibody and the short hairpin complex, or the single controls. So after the treatment, we co-culture them with PBMCs and then measure interferon gamma production. And you can see how this production was just occurring in the cells that were treated with the TMAF and the short hairpin. When we repeated these experiments, but with the KPCs that were RIG1 and ENT2 knockout, we see that this production is abolished, meaning that we can take advantage of this system that has like high expression of ENT2 to trigger and change the tumor microenvironment. So I hope that today you see TMAV as a novel, potent, and specific tool to deliver therapeutic RNAs. And in our case, when we pair it with this short hairpin, it enhances animal survival and increase the activation of TCD8 cells, modifying the tumor microenvironment and therefore increasing animal survival. So I just want to thank my lab, Dr. Escobar Hoyos, and our collaborators for making us part of this project and also for our funding. Thank you.
Thank you, Dr. Martinez Soto. Next, our speaker will be Dr. Shin Hamada. Dr. Shin Hamada is a senior assistant professor at Tohoku University. Today, Dr. Hamada will be speaking to us about stress response mechanism and regulation of inflammatory molecules in pancreatic cancer. Dr. Hamada.
Okay, so it is a great pleasure for me to present our work at this meeting. So I would like to talk about the regulation of inflammatory molecules by the oxidative stress response mechanism in pancreatic cancer. So there's no conflict of interest to declare. The KEAP1 NR2 system plays crucial roles in the cellular responses to oxidative stress. Under normal condition, the adapter protein KEAP1 binds to NRF2 and facilitates degradation by proteasome. However, increased oxidative stress attacks disulfide bonds in the KEAP1, which leads to the conformational change. This attenuates degradation of NRF2. Then NRF2 translocates to nucleus and increases expression of cytoprotective genes. This process contributes to tissue homeostasis and protection. However, activation of oxidative stress response in cancer cells yields growth advantage and chemoresistance. KPC mouse is an established pancreatic cancer model. Pancreas-specific expression of constitutively active KRAS and mutant P53, which acts as dominant negative, cause invasive pancreatic cancer, resulting in death within 1 year after birth. Tissue structure of pancreatic cancer in KPC mouse recapitulates human pancreatic cancer, represented by fibrosis and inflammatory cell infiltration. The previous report suggested increased oxidative stress by mutant KRAS expression in the mouse model. Deletion of NR2 in mutant KRAS expressing mouse led to the increased reactive oxygen species level in the pancreas. So to validate whether NR2 contributes to pancreatic carcinogenesis in KPC mouse, we introduced NR2 null background as shown in this slide. As a result, NR2 deletion reduced the number of panning the precancerous region, both in mutant KRAS and mutant KRAS P53 expressing mice. Furthermore, NR2 deleted KPC mouse developed less invasive pancreatic cancer compared to wild-type KPC mouse. KPC mouse developed liver metastasis, lymph node metastasis, direct invasion to surrounding organ. However, pancreatic cancer developed in NR2 deleted KPC mouse remained within the pancreas. And panning lesions developed in mutant KRAS and mutant KRAS P53 expressing mice were accompanied by inflammatory cell infiltration. And deletion of NR2 also decreased inflammatory cell around panning. So as reported previously, inflammation accelerates pancreatic carcinogenesis. And we carried out following experiments to clarify whether the KEAP1 NR2 system regulates inflammatory molecules during pancreatic carcinogenesis. So as a method, pancreatic cancer cell lines were established from KPC mouse and NR2 deleted KPC mouse. And immortalized pancreatic stellate cells were established from wild-type and NR2 knockout mice. Gene expression profiles were compared by microarray, and expression of inflammatory molecule was assessed by Western blot and immunohistochemistry. At first, we established pancreatic cancer cell lines from KPC mouse and NR2 deleted KPC mouse. NR2 deleted KPC mouse derived cell lines lack nuclear NR2 expression. And the expression of NR2 target molecule, NQ1, was also lost. Ductal molecule CK1719 was expressed to similar extent. GH8 is the typical NR2 inducer that attacks KEAP1. And them treatment significantly decreased cellular viability of NR2 null pancreatic cancer cell lines. So this data confirmed vulnerability to oxidative stress by NR2 deletion. And gemcitabine is a standard chemotherapeutic agent used for pancreatic cancer. And gemcitabine treatment also significantly decreased cellular viability of NR2 null pancreatic cancer cell lines. So this result suggests that NR2 correlates with sensitivity to chemotherapy. And to further explore downstream genes affected by NR2 deletion, we performed microarray. Glutathione S transferase family genes, ABC transporter, and UGT glucuronide transferases were found to be downregulated. In addition to these genes, IL-33 was also downregulated. IL-33 belongs to IL-1 family, and IL-33 is released by tissue damage and stress, causing activation of inflammatory signals. And recently, oxidative stress increases IL-33 release from airway epithelial cells. So we validated IL-33 expression in NR2 null cancer cell lines and wild-type cancer cell lines. NR2 deletion decreased IL-33 messenger RNA expression significantly. Also, nuclear expression of IL-33 was also reduced in NR2 null cancer cell lines. So NR2 deletion in pancreatic cancer cells caused vulnerability to oxidative stress and chemotherapeutic agent, and loss of NR2 in pancreatic cancer cells decreased IL-33 expression. We next established immortalized pancreatic stellate cell lines from wild-type and NR2 knockout mouse. These cells were immortalized by SP40 introduction. And as you can see here, NR2 expression is lost in NR2 null PSG cell line. But these cells grew normally under normal condition, similar to wild-type PSC line. And similar to NRF2 neuropancreatic cancer cell lines, cellular viability of NRF2 neuro-PSC line significantly decreased by DEM treatment or another oxidative stress-induced hydrogen peroxide compared to wild-type PSC line. And microarray analysis identified decreased expression of NRF2 target molecules such as glutathione peroxidase 3, NQ1, and ABCC2. And together with genes, IL-33 and ICAM1 were downregulated in NRF2 neuro-PSC line. We also validated IL-33 and ICAM1 expression in NRF2 neuro-PSC line and wild-type PSC line. NRF2 deletion decreased IL-33 and ICAM1 mRNA expression significantly, and also nuclear expression of IL-33 and ICAM1 expression were also reduced in NRF2 neuro-PSC line. So NRF2 deletion did not affect cellular growth of immortalized PSCs, but loss of NRF2 caused vulnerability to oxidative stress. And NRF2 was involved in the expression of inflammation-related molecules in PSCs. Since NRF2 has pivotal roles in various cancer, several inhibitors of NRF2 have been developed. And a recent study established a monitoring cell line for NRF2 activity for high throughput analysis. And this approach identified the febrifigin plant alkaloid as a candidate for NRF2 inhibitor. And derivative of febrifigin, halofuscinone, rapidly depleted NRF2 in the esophageal squamous cell cancer cell line and the lung cancer cell line with constitutive NRF2 activation. And halofuscinone showed anti-cancer activity in cell lines. So we assessed whether halofuscinone has anti-cancer effects in pancreatic cancer cells. Treatment of KPC mouse-derived cancer cell lines with halofuscinone resulted in NRF2 depletion accompanied by EIF2 alpha phosphorylation, a hallmark of amino acid depletion. And halofuscinone treatment sensitized KPC cell lines to gemcitabine. We also evaluated the efficacy of combination therapy in subcutaneous implantation model. As shown in this slide, addition of halofuscinone treatment to gemcitabine further decreased subcutaneous tumor size compared to gemcitabine monotherapy. So administration of halofuscinone to KPC mouse also attenuated cancer progression. Intraperitoneal injection of halofuscinone for 4 weeks significantly reduced number of pannings in KPC mice. And immunohistochemistry for IL-33 detected IL-33 positive panning-forming cells and panning-surrounding cells in KPC mouse. And as shown in this slide, 4 weeks of halofuscinone treatment reduced inflammatory cell infiltration around panning in KPC mice. And halofuscinone treatment also decreased IL-33 positive panning-forming cells and surrounding cells. So in summary, the NRF2 inhibitor halofuscinone caused rapid depletion of NRF2. And halofuscinone treatment reduced panning formation and inflammatory cell infiltration. So halofuscinone treatment also decreased IL-33 expression. In conclusion, IL-33 expression was regulated by NRF2, both in pancreatic cancer cells and PSCs. And deletion or pharmacological inhibition of NRF2 reduced inflammatory cell infiltration in the microenvironment of pancreatic cancer. As a future direction, contribution of IL-33 during pancreatic cancer development needs further validation, and cell type specific deletion of IL-33 might be adequate approaches. So thank you for your attention.
Dr. Hamada. Our next speaker is Dr. Ming Yang Lu. Dr. Ming Yang Lu is a researcher affiliated with Peking Union Medical College, Cancer Hospital in Beijing, China. Dr. Liu's research primarily focuses on the role of tumor microenvironment in pancreatic cancer cachexia and chemoresistance. Dr. Liu will be speaking to us about enhancing the chemoimmune therapy response of pancreatic cancer by targeting tumor immune microenvironment. Dr. Liu?
Okay, good morning, everyone. Thanks for the introduction. First, I want to thank Professor Saluja and Dr. Saluja and Dr. Lee's invitation. And I'm happy and honored to present my recent research on tumor immune microenvironment and pancreatic cancer progress. First, I will show you the background. Pancreatic cancer is one of the malignant gastrointestinal cancers, and it's the third leading cause of cancer-related deaths in the United States. And only 20 to 30% of the patients have the chance to receive the surgical resection, and many patients are tolerant to either chemo or immune or targeted therapy. So the 5-year survival for the patients is only 13%. And recently, we focus on the tumor microenvironment for the pancreatic cancer, which is characterized by the suppressive tumor immune microenvironment consistent with the suppressive immune cells, such as M2 macrophages, Th2 cells, tolerogenic DC cells, and Treg cells. So it's reported that the suppressive immune microenvironment promotes the pancreatic cancer progress. And we focus on a new phenotype called cancer cachexia, which is a chronic syndrome of wasting. And for the pancreatic cancer patients, almost 70 to 80% of patients will develop the cancer cachexia during the process of the disease. And almost a third of the patients will ultimately die because of the cancer cachexia. And the clinical symptoms of the cachexia include more than 5% loss of weight, and the patients will have muscle loss with or without adipose loss, and the patients will have severe emaciation, decreased appetite, and nausea. Since we don't know the correlations between the immune microenvironment and the cancer cachexia, so first we analyze the correlations between the immune microenvironment and the cachexia prevalence. We found that the macrophages, they are positively correlated with the cachexia prevalence and as well as the weight loss for the patients. And we also found the pancreatic cancer patients with high macrophage percentage, they have poor survival. And next, we analyzed the gene signature of the immune cells. We found in the macrophage, the gene signature is enriched in the cachexic tumor tissue comparing with the non-cachexic tumor tissue. And now we did some gain loss functional study, and we found in the cachexic tumor tissue, there are more macrophage infiltrated. And now we treated the muscle with the cachexic cancer cells and co-cultured with the macrophage. We found that the cachexic cancer cells treated with macrophage, it can induce the muscle wasting significantly. And the mechanism part we found in the cachexic cancer cells, we can see more the chemokine CCL2, which can recruit and activate the macrophage. And overall, the mechanism part, we found the cachexic cancer cells, they can secrete more CCL2, which can recruit and activate macrophage. They in turn secrete more chemokine CCL5 and activate the signaling pathway in the cancer cells with a higher polyubiquitination of Child-6 that activate the phosphorylation of downstream kappa B, P65, and transcriptionally activate the TWIC, which can secreted by the cancer cell and bind to the receptor for the muscle and then activate the signaling pathway in the muscle to induce the muscle wasting. And this part of work was done in my mentor, Dr. Min Li's lab, with a great help of my colleague in Dr. Li's lab. And now I will talk about my recent work on the chemo resistance and immuno invasion. Since only 20 to 30% of pancreatic cancer patients, they have the chance to receive the surgical resection, and most of them, they can only be treated with either chemo or targeted therapy. So gemcitabine is one of the first line drug for the chemo treatment. But many patients, they are tolerant to gemcitabine treatment. One mechanism is because of the cell intrinsic resistance. That means gemcitabine needs to be transported with the transporter named ENT1 to deliver into the cell. We found in the resistant cancer, in the resistant patients, the tumor tissue, we see less ENT1 expression. And at the same time, it's reported that the catalyzing enzyme named DCK is inactivated in the resistant patient's tissue, which can inactivate gemcitabine. And it's also reported that in the resistant patients, there are more glycolysis level, which can produce more the metabolic, such as the deoxycytidine, which can compete with gemcitabine to enter into the nuclear and induce the gemcitabine resistance. And the other part for the gemcitabine resistance is the non-cell autonomous resistance. That means the tumor cells can interact with other, such as immune cells, to activate the signaling pathway in the cancer cells to induce the gemcitabine resistance. And it's found that the tumor-associated macrophages, it can induce the chronic inflammation in the cancer cell, as well as suppress the adaptive immunity and induce the EMT to induce the gemcitabine resistance. And it's reported that the tumor-associated macrophages, it can inhibit the activity of the catalyzing enzyme, such as DCK, which inactivate gemcitabine in the cancer cell. And they found co-cultured with the macrophage with the cancer cells, it will produce more the metabolic, such as cytidine, to compete with gemcitabine. And they did the immunofluorescence study and found if they knock out CCR2, which is the macrophage marker, if they inhibit the macrophage, they can see more gemcitabine entered into the tumor tissue. And for the function of gemcitabine and the immune response, it's found that one part gemcitabine can suppress the immune microenvironment by promote the macrophage, such as M2 macrophage growth, as well it induce the MDSC infiltration. But at the same time, gemcitabine can also stimulate the immune microenvironment by inhibit the Treg, decrease the Treg population. And the long-term treatment with gemcitabine may reprogram the tumor microenvironment. But gemcitabine in itself, it didn't change the T or B lymphocyte in the tumor tissue. But if combined gemcitabine with the PD-1, anti-PD-1, and some targeted therapy, such as TGF-beta, it's found the triple combination treatment, it decreased the tumor burden, overlong the survival for the mice, and increased the response to the immuno treatment. And they found it increased the CD8 infiltration. So that means gemcitabine, it inhibits the immune checkpoint by enhancing the antigen presentation, only following the disruption of the immunosuppressive cytokines barrier. So next, we want to investigate such kind of targets for the targeted therapy. So first, we establish the gemcitabine resistant cell lines by continually treating the cancer cell with gemcitabine. And we found in the resistant cell lines, we can see a much higher IC50 of gemcitabine. And as well, we investigated the function of the resistant cells on the CD8 T cell. We found in the resistant cells, they can decrease the CD8 T cell function by inhibiting the recruitment of CD8 T cell, inactivate CD8 T cell with the inhibiting the activation marker of CD8 T cell. So next, we want to know the mechanism. So we did the RNA sequencing with the wild type and the resistant cell lines, and we found 6 targets. One of them is ZEP1. When I was the postdoc in my mentor, Dr. Minley's lab, we found ZEP1 can enhance the gemcitabine resistance through the integrin alpha-3 beta-1, inhibit the ENT1 activity to induce the gemcitabine. And we found ZEP1 is upregulated in our resistant cell lines. And next, we knocked down ZEP1, and we found it decreased IC50 for gemcitabine and increased the recruitment for CD8 T cell and activate the CD8 T cell with a higher population for the interferon GCMA and GCMB, which are the activated marker for CD8 T cell. And next, we want to know that whether knockdown ZEP1, it increased the gemcitabine resistance, whether it's a contribution with the tumor immune microenvironment or not. So we injected the ZEP1 knockdown cells both into the immune competent and immune deficient mice. And we found only in the immune competent mice, knockdown ZEP1 significantly decreased the tumor growth, increased the gemcitabine resistance. So next, we established the orthotopic allograft mouse model with ZEP1 knockdown and combined ZEP1 knockdown with the gemcitabine. And we found the treatment with gemcitabine plus ZEP1 knockdown, it significantly decreased the tumor growth, increased the gemcitabine resistance, and it prolonged the overall survival of the mice. And next, in order to know that how does knockdown ZEP1 affect the tumor immune microenvironment, so we collected the tumor tissue and did the single cell RNA sequencing. And we found in the red panel, we found knockdown ZEP1, it increased the population of the T cells as the green color indicated. And next, we did further subgroup analyze and found it knockdown ZEP1, it increased the population of the infected CD8 T cell, significantly increased the percentage. And next, in order to investigate that the spatial change for the especially CD8 T cell, so we did the spatial transomic screening, and we found comparing with the control group, we found knockdown ZEP1, it significantly increased the CD8 T cell infiltration, especially we can see in the tumor central part, we can see more CD8 T cell. And we did the interaction, we did the crosstalk analysis, and we found knockdown ZEP1 in the tumor tissue significantly increased the GCMA positive CD8 T cell infiltration. And next, we verified the findings with the multiplex immunofluorescence. We stained the tumor cell with the CK19 as the red color indicated, and we also stained the CD8 with the green color. We can see in the ZEP1 knockdown group, we can see more CD8 infiltrated. So that means knockdown ZEP1 is significantly activate the tumor immune microenvironment by increase the GCMA positive CD8 T cell infiltration. And next, we did some functional study. We found knocking down ZEP1, it significantly increased the CD8 T cell recruitment, activated the CD8 T cell, decreased the CD8 T cell apoptosis. And we verified the findings with Western blot. We found that knocking down ZEP1, it decreased the apoptotic markers such as cleave the caspase-9 and caspase-3. And since CD8 T cell function is characterized by the cytotoxicity, so we established the CAR T cells with the antigen vector overexpressed in the cancer cells. And also we collected the OT1 CD8 T cell from the mice and also constructed the antigen-based vector overexpressed in the KPC cell. And we found knocking down ZEP1, it increased the cytotoxicity of the CD8 T cell. And next, we did some in vivo study to verify our findings. And we found knockdown ZEP1 combined with treatment with the OT1 CD8 T cells through IV. And we found it significantly inhibited the tumor growth, increased the gemcitabine response. And next, since gemcitabine need to be transported into the cells through the transporter ENT1, so we measured the ENT1 expression in the cancer cells with ZEB1 knockdown plus treated with the CD8 T cell. We found knockdown ZEB1 it a little bit increased the ENT1, but if we knock down ZEB1 plus treatment with the condition medium from CD8 T cell, we found it significantly increased the expression for the ENT1. And next, we did the immunofluorescence staining, and first we stained the CY5 to label gemcitabine. And we found knockdown ZEB1 plus CD8 T cell treatment, we found it significantly increased the gemcitabine signal in the cells. And also we investigated the ENT1 level. That is consistent. That means a knockdown ZEB1 plus CD8 T cell treatment, it increased the ENT1 level in the cells. And next, we combine the ZEB1 knockdown and the immune checkpoint and the PD-1 and plus gemcitabine. And we found the triple treatment significantly inhibited the tumor growth in the orthotopic mouse. And next, we did the clinical relevance study. We stained the ZEB1, CD8 in the pancreatic cancer patient specimen. And we found in the gemcitabine-resistant patients' tumor tissue, there are more ZEB1 staining, but less CD8 staining. But in the sensitive tumor patient, the sensitive patient's tissue, we can see less ZEB1 staining, but higher level for the CD8 T cell infiltration. So to summarize, we found the macrophages can promote the pancreatic cancer caucasia, and targeting the EMT transcription factor ZEB1 can activate the tumor immune microenvironment and sensitize the pancreatic cancer to the chemo and the immunotherapy. And ZEB1 inhibition it can enhance the anti-tumor response of CD8 T cells. And for the clinical relevation, we found ZEB1 is positively correlated with gemcitabine in pancreatic cancer. So now finally, I want to thank my Dr. Min Li, who gave me great help and support and guidance all the time. And I also want to thank my colleague in Dr. Min Li's lab, including Jingxuan Yang, Zhijun Zhou, Yu Ren, and Yang Cai, who contribute a lot to that cancer cell, the caucasia paper. I really appreciate their help. And I also want to thank my colleagues in the POMC Cancer Hospital, including my director, Zhihua Liu, and my postdoc and my PhD student who contribute a lot to that ZEB1 paper. And finally, I want to thank my collaborators in China, including POMC Cancer Hospital, Shanghai Regional Hospital, and Xi'an Jiaotong University, and the Peking Hospital. And finally, I want to especially thank Dr. Shrak Saluja, my career mentor, for his support and the guidance. I also want to thank APA for the invitation. Okay, that's all. Thank you.
Thank you, Dr. Lu, for that excellent talk. Now we'd like to invite all of the speakers up to the podium here to sit next to us. We'll open the next portion for the next maybe 10 minutes or so for questions and discussion regarding these great presentations.
Please.
Can we turn that mic on, please?
Hi, there we go. Hi. Hi, I'm Julie Carsons from the University of Alabama at Birmingham. So I have a question I'd really like all the panelists to be able to address. You did a lot of amazing work impacting the tumor microenvironment on primary tumor progression. What about the metastases? Did you assay it? What was different? Where did it go? You know, were these therapies effective in that environment as well?
Okay, so hello, thank you for your question. So in our case, that is something that we are interested in because, you know, we know the issue that is metastasis. So right now we are working in establishing models and see how this treatment is going to behave, how the antibody and the short hair period are going to behave in that. But it's something that we are still working on, and that we are aware that we need also to tackle and solve. Yeah.
Okay, we are mainly interested in the resistance mechanism of pancreatic cancer to various stimuli such as chemotherapy or immune cells. And according to our experience, I'd like to say such dependence to stress response mechanism during carcinogenesis is highly context and tissue specific, because we could observe inhibitory effect of NR2 deletion in pancreatic carcinogenesis. But in case of cholangiocellular carcinoma model, by using albumin-free mice, by crossing them with KRAS and P53 mice, we can develop cholangiocarcinoma. However, in that situation, NR2 deletion has no additional meaning. In contrast, deletion of KEAP1, which can cause constitutive activation of NR2, can enhance carcinogenesis. So, it's so highly context dependent, but in each cases, stress response mechanism has specific role. So I'd like to confirm some good therapeutic target in that context.
In my opinion, I think in order to cure the pancreatic cancer, to decrease the tumor burden, I think we need to increase the chemo and the immune response for the patients. As I just mentioned, gemcitabine itself is not enough. They need to combine with the immune checkpoint and plus the targeted therapy to activate the immune microenvironment. Because gemcitabine in itself can't induce the cytokine storm. You need to combine with the targeted therapy to activate the immune cells, such as CD T cells, to induce the cytokine storm to better enhance the response of the pancreatic cancer patients to the clinical treatment.
Hey, Julie. So in my studies, I mainly focused on the primary tumor progression because, as you know, that I use the acute mouse model with the homozygous P53 loss. So it saves us a lot of time as a new lab. But on the other hand, it cannot mimic like KPC model, the traditional R172H mutation, and to have sufficient metastasis to study it, right?
Doing a follow-up to that, Yang, with your work on the myeloids and how important the myeloid is also in the liver, do you have a guess of what you think might be going on?
Yeah, so when I started, I based on what the data, the single cell data we established in Dr. Kaluri's lab, I did investigate, combine all the data set we had, the KBC primary tumor, later stage, like primary, early stage, late stage, and then liver metastasis, to characterize the tumor microenvironment changes across the different stages with a very simple computational study. We did see some similarities and differences regarding that, but you can check. I can share with you the link. Thank you.
Hi, thank you for your really nice talks. I have a question for Dr. Chen. Thank you very much for having this model, actually, because I think it has to be underlined as one of the first, together with the KIT M6A knockout that shows adenosquamous tumors in the mouse. So we can use it now as a model, which is really great. One of the questions is related to the fact that you're using PDX1-3, so you have recombination in the embryonic pancreas. And you know that there's differentiation coming and then this big proliferative phase. So my question is, did you try actually to conditionally recombine these alleles in the adults in specific cell types and see what happens there?
Definitely, yeah, definitely. Because like we considered using different Cree lineages, including the inducible Cree ERT2 or different Cree like elastase Cree, for example, or SOX9 Cree ERT2, things like that. But in the beginning, we were not even sure whether it will generate tumor at all, right? But for the future studies, I think it's worth it to study using different Cree lineages, inducible or not, to study that.
And one of the questions is also related to when does the tumor happen to evolve and to
Right, right.
develop? Is it like
Sure,
after
it's
weaning?
much slower. It is much slower. So, for example, the best control we had, because we only have the KRAS mutated control models. So we use the one with the KRAS mutation and P53 homozygous loss with the same Cree, and then we compare it to the P53 SMAD4 homozygous loss with the same Cree. And of course, the KPPC mice will die around like 3 months, and then the PPSSC or PPTTC, these mice will usually die around from 9 to 12 months of age. So it's much slower model.
But when does it start to develop?
So we checked early stages, for example, like 6 months to around 6 months. So they start to develop metastatic lesions around that age.
Thank you. And then last question, did you compare with human adenosquamous tumors? Like how does the P63, for example, expression relate to that? Because
Right,
we know
right.
you have nests. Usually the P63 is at the edge of these nests, and inside you have other kind of cells.
Right, we haven't looked into that because of the lack of samples with the squamous subtype. Yeah.
Okay,
But
thank
it's
you very much.
important. Thank you.
Yeah.
It's not a question, but more of a programmatic announcement that, first of all, thank you for all of you for staying. I know all of you are saturated after 4 days of meetings, but after the next session, please make sure you stay for lunch, which will be served immediately after that. So thank you again for all the brave souls who are still here. Thank you.
Thank you. Session again at 11:30.